Hey guys, welcome to the first video of my YouTube channel. Today's topic is understanding the concepts of allele and different types of monogenic interactions. In the subsequent video, we will discuss about the two-gene interaction and polygenic interactions. Before we start with the alleles, we should understand the difference between the terms character, trait, unit factor or gene, and allele. Character is the feature of the individual, like height, color, or shape. Whereas trait are the detectable and contrasting variant of the inheritable character. As for example, if we take height as a parameter, then tall and dwarf are the traits. Now comes gene, which is previously known as unit factor. The term gene was coined by Johansen. He has also coined terms like genotype, phenotype, and pure line. Gene contains information to express a particular trait. If one gene controls one character, then it is known as monogenic interactions. As for example, incomplete dominance, codominance, multiple allelism, lethality, and pleiotropy. Do you know the difference between allele and gene? Allele is a short form of allelomorph, which means form of one another. The term allele, homozygous and heterozygous were given by Peterson, the father of modern genetics. So the allele is the alternative and contrasting form of the gene, which are responsible for contrasting trait of same character. For example, yellow seed color is given by capital Y allele, no matter whether it is present in homozygous condition or heterozygous condition. And alleles are located in the same relative position, which is also known as locus, of homologous chromosomes. An allele could be dominant or recessive. In other words, we can say in any diploid organism there are two copies of gene, and they are nothing but the alleles. These alleles may be identical or non-identical. Let's assume allele capital T codes for some specific kind of protein which increases the plant height, and it has undergone a kind of mutation. Now three conditions could appear. Condition 1, in which normal and functional protein is produced. And here the allele acts as an unmodified allele, thus expressing the original phenotype and known as dominant allele. In condition 2, non-functional protein are produced, and in condition 3, no protein is produced. In both the cases, there will be no increase in the height of the plant. Thus, it's a recessive allele. Now, there is also a fourth chance of production of altered but functional protein, which may give rise to conditions like incomplete dominance or codominance. Incomplete dominance. As the term refers, no trait will be completely dominant, and it will be a fusion of inappropriate dominance and creation of an intermediate product. The examples to be noted are mirabilis and antirrhinum. Mirabilis is also known as 4 o'clock plant, and another name of antirrhinum are snapdragon or dog flower. In both the cases, we can see red is the dominant flower color and white is the recessive flower color. But there is generation of intermediate pink color flower in the heterozygous condition. Such instance may be explained as because of the formation of altered and functional protein due to modified allele, as we have discussed earlier. Codominance. As the term refers, there must be coexistence of dominance of both contrasting traits. That means both traits can express themselves, but not to a full extent. To understand this, we can have the example of roan cow and blue and white bird. As we can see, there is no expression of single color, but both the colors are mosaically expressed. In human, codominance can be found in heterozygous condition of sickle cell anemia, AB and Mn type of blood grouping. Now in both incomplete dominance and codominance, the phenotypic and genotypic ratio comes to be 1:2:1.